was a very interesting panel at ARM. It's always a great meeting to discuss with colleagues and leaders of the field of regenerative medicine, and in particular cellular therapy. What we discussed during that panel is really around what progress are we making in different companies in terms of addressing the needs in hematological malignancies, liquid tumors, as well as in solid tumors. We also discussed a recent event related to safety of cellular therapy, because some particular gene editing technology seems to have some trouble in terms of inducing some chromosomal abnormalities in very few patients there. And that's creating some question mark on some technologies. And then we also discuss between allogeneic cell therapy and autologous cell therapy, whether there is now a clear winner, if I may say, in terms of the best technology for patients and patient access there. So if I come back to the liquid and solid tumors, what we discuss more precisely is what is the medical need that should be addressed by new generation of cellular therapy in liquid tumors. And we believe at Atara that there is a medical need in terms of having deeper response rate, so higher level of response rate, as well as longer durability of remission, because persistence of the cell therapy, especially for allogeneic cell therapy, and the link with the durability of remission is still an unmet medical need. And then, of course, on safety, where we need to ensure good safety for the patient, even better, hopefully, than the autologous CAR-T, autologous cell therapy there. So we are working at Atara with that, especially with the CD19 allogeneic CAR-T that we are developing. Now, on the solid tumors, there are different approaches to try to address the unique specificity of solid tumors in terms of how to make cellular therapy as effective in solid tumors as they are in liquid tumors. And one of the paths that Atara is choosing is to improve the functional persistence of the cells with our allogeneic CAR-T program, and we have a program targeting mesothelin for solid tumor. And here it's how do we develop a construct of the cells with new co-stimulatory domain. The one we use is 1XX, coming from Memorial Sloan Kettering and invented by Michel Sadelain, which is a co-stimulatory domain that avoids to exhaust the T cells, so you have more persistence. And then including into the construct of the CAR-T a PD-1 dominant negative receptor that allows to address the immunosuppressive environment and allow the CAR-T to be more functionally persistent, which means to be able to not only stay longer into the tumor, but be able to expand and eradicate the tumor cells when exposed to the antigens. And we've had great recent preclinical study being presented last year at various congresses that shows that this type of construct, under the name of ATA2271 or ATA3271, are really extremely helpful in addressing this particular challenge of solid tumors, and having the ability for this allogeneic CAR-T 3271 to expand and eradicate tumor on a multiple tumor rechallenge, which means that it is more functionally persistent. The cells are still active for a longer time. And that's, we believe, extremely important to address the needs of solid tumors treatment.